Okay, welcome to Concept 2 Notes, where we're going to be talking more about fertilization. So far in Concept 1, we've talked about the overall structures in the male and female reproductive systems and then their roles in reproduction, mainly through our discovery stations. And in the notes, we highlighted some key events in the reproductive cycles for females, as well as puberty in both male and females. But we haven't actually talked about how it actually all goes down. How is an offspring actually made? And that's what concepts 2 and 3 are going to be all about. So, in this concept, we're going to review mitosis and meiosis from biology 1 before we're kind of going to jump in to how eggs and sperm are specifically made. And then we're going to kind of finish with the sperm fertilizing the egg. And end with the zygote. And then in concept 3, we're going to pick up with that zygote and how it develops into an embryo, which develops into a fetus, which will hopefully be birthed as a healthy infant at the end of pregnancy. So here we go. Overview. Look at those cute babies. Babies can't happen without pregnancy, and pregnancy cannot happen without fertilization. And fertilization cannot happen naturally without copulation, which is sexual intercourse. Now, of course, fertilization can happen artificially with in vitro fertilization, which we'll be talking about in class, but there has to be this joining process. And then copulation can't result in an offspring without egg and sperm. So where are egg and sperm coming from? That's kind of our essential question of the day, if you will. All right, so let's review. Meiosis. This is the process of cell division that's making gametes, and it's happening in the gonads. So males are making sperm in their testes. Females are making eggs in their ovaries. This process makes haploid sex cells. They have half the number of chromosomes as all the rest of the cells in your body, those diploid body cells. So haploid just means one full set of chromosomes, which is 23 in humans. Diploid is 2 full sets of chromosomes, which is 46 in humans. Sexual reproduction can't happen without meiosis. Okay, so we've got to make these cells that have half the number. So if I have a half and a half, when they fertilize, then I get a whole. And then that fertilized egg is going to go through mitosis, which we'll talk about in a minute, and differentiation to get to that embryo. But let's talk meiosis. Okay, so hopefully this is all a review from Biology 1. I'm not going into a lot of detail on this because you should remember. So if you don't, that is something important. I've linked a video that you can watch that'll kind of reteach you this from Biology 1. But essentially, let's say we start with a cell, a parent cell that looks like this. So let's count those chromosomes. Note there's 1, 2, 3, 4. Again, in a human, this would be 46. Okay, our body cells have 46 chromosomes, but that would be a much more complicated diagram. So we're just going to show these an organism that's diploid number is 4. Okay, so we have simplified our cell drawing. We basically just have a cell membrane, a nuclear membrane in our nucleus with our nucleolus and our chromosomes. Then we got some centrioles with some spindle fibers coming out of them. Okay, so we're just focusing on the main things here. So starting here, okay, in meiosis, first, those chromosomes pair up. So chromosome number 2 from mom and your chromosome number 2 from dad get together and vice versa. So you would have 23 pairs here. Here we only have 2 pairs. But again, still 4 chromosomes. Then the S phase of interphase happens, which is DNA replication, synthesis. We double those. Now we have sister chromatids. So now I have 2 copies of each one. All right, and then we enter into prophase, metaphase, which is where those pairs, metaphase 1, they line up in the middle of the cell together. Anaphase 1, those pairs separate. And then here, these 2 are showing telophase 1, which is where we're starting to form 2 different cells. And then we have cytokinesis. Okay, and notice at the end of cytokinesis, I have 2 cells. They only have 2 chromosomes in them. We started with 4. Now we have 2, but we still have duplicates of each one. Okay, so we're down to haploid, but we still have duplicated chromosomes. We still have those sister chromatids. So we do it all over again. Prophase 2, we've got metaphase 2 where they line up in the middle of the cell. We have phase 2 where we start separating those. And then telophase 2 and cytokinesis, where you can see our end result here is we end up with 4 cells. Okay, we started with 1, we end with 4. And notice, each only has 2. We started with 4. So we end up with half the number of chromosomes that we started with. That's so important in meiosis. Okay, so this is how we would get 4 sperm. In females, we actually only end up with 1 that would actually be considered an egg. The other 3 are polar bodies that degenerate, which we'll talk about. But this is how we're getting gametes here. So then how does this happen? How do we have, a baby on an ultrasound, and then we get this baby, and then this baby grows up into an adult? How is that going down? Well, that's going down because of mitosis, which is the process of cell division that's making identical copies of our somatic or our body cells. So we're making diploid cells. We're copying all of your genetic information. Diploid is 2 full sets of chromosomes, or 46 in humans. So those gametes we made in meiosis, they fertilize. We now have, our first time we have all 46 chromosomes. That fertilized egg is going to go through mitosis repeatedly and just make a clump of identical cells. But then those identical cells start to differentiate. They differentiate into specific tissues, into organs, and then organ systems that are eventually going to make up the embryo as an organism. Again, this should all be a review. Okay, so in mitosis, we start an interphase. You may remember the DNA is kind of like decondensed, and so we refer to it as chromatin. But then those chromosomes kind of condense. The S phase of interphase starts again. So we have the duplicated chromosomes. We have 4 here. They line up in the middle of the cell in metaphase. Then they get split. We've got telophase or anaphase happening here, and then telophase. And then note we're going to end with 2 cells that are identical to what we started with, and they're each going to have 4, which is what we started with as well. So we're going to start and end with the same number here. We're making identical copies of cell. So 1 cell becomes 2 with the same numbers of chromosomes. So we start with 4, we end with 4. In humans, we start with 46 and we end with 46. All right, so let's just do a basic overview. We have mitosis, which is the creation of diploid somatic cells. This is going on throughout your life and throughout your whole body. And its main purpose is just growth and repair. PMAT, that prophase, metaphase, anaphase, telophase, just happens 1 time, and it makes 2 identical diploid somatic cells. And this whole process is asexual. You do this all by yourself. Meiosis makes haploid sex cells. In females, it starts before you're born. Okay, males, it's going on throughout your whole life from puberty on. And it's going on in your gonads, your ovaries, or your testes, depending on if you're male or female. The purpose is to make babies, and we do PMAT twice. We go through prophase 1, metaphase 1, anaphase 1, telophase 1, and then we do that all over again a second time. And this is how we're able to get 4 unique haploid gametes as a result. And it's a sexual process. To get a baby, that's our end goal. It takes a male and a female, and it takes that fertilization. All right, so in class, we're going to pause and just review this because I know that this is a lot and you may not remember because it may have been a year or 2 since biology 1. But for the sake of the video, we're going to keep going through. So let's talk specifically about spermatogenesis. This is the process of forming male gametes. So we're doing meiosis in males. It's happening in the seminiferous tubules of the testes. Those are known as the sperm factories, if you remember from the discovery stations. And it's going down at puberty, which is usually around 14 years in males, but that can vary. And the release of testosterone during this time is what's causing the process to start. And then testosterone is also what's driving this process throughout the rest of a male's life. And so we're going to take 1 spermatogonium cell, and it's going to make 4 spermatids. So 1 spermatogonium cell, and we're going to end up with 4 spermatids that will develop into 4 mature sperm. So those spermatids go through spermiogenesis, which is the process that makes them mature and makes them motile, makes them able to essentially swim. So prior to puberty, mitosis of stem cells, which are your spermatogonia, which is plural, or the spermatogonium, which is singular, is going down in the seminiferous tubules. And we're just making more and more identical spermatogonia daughter cells. But then once puberty hits, then some of these are going to remain as stem cells, and we're just going to keep doing mitosis and making more of these stem cells, spermatogonia. But then some are going to go on and actually go through meiosis, and that's where we're going to get the sperm. A healthy male can make 90 million sperm a day. That's a lot of sperm. And there are a bunch of other important cells in the seminiferous tubules. There are cystanocytes, which are supportive. They have supportive roles in sperm formation. There's spermatogenic cells, which are going to give rise to sperm, like I was just talking about, those germ cells. There's myoid cells, which are smooth, muscle-like cells that are rhythmically contracting to help squeeze the sperm out of the tubules once they're made. And then there's interstitial endocrine cells, also known as Leydig cells, that are producing androgens, most importantly testosterone, to kind of keep everything going. So spermatogonia, okay, those stem cells are going through mitosis. They're just making identical copies of themselves until puberty. After puberty, mitosis is going to make type A daughter cells that are going to just keep dividing, keep doing mitosis, keep making spermatogonia, so that we're always going to have these cells to make more sperm. But then there's going to be these type B daughter cells, which are going to become primary spermatocytes that are going to then undergo meiosis and become sperm. And then again, like I mentioned, spermiogenesis is what's maturing these non-motile spermatides into mobile sperm. So again, if we start with 4 chromosomes, S phase of interphase happens. We have now 4 duplicated chromosomes. Meiosis 1 goes down, we get these secondary spermatocytes, and then meiosis 2 goes down and we get these 4 spermatids, which will then be matured in spermiogenesis into 4 sperm. All right, let's talk about the structure of a sperm really quick. There are 3 main parts. The head, which is the genetic region. So structurally, we call it the head. Functionally, it's known as the genetic region. It contains the flattened nucleus, which has all the DNA in it. And it's surrounded by this acrosome, which secretes hydrolytic enzymes. And those are really important for when the sperm gets to the egg so that it can kind of penetrate the egg's membrane and actually fertilize it. All right, the middle is known as the midpiece. Structurally, functionally, it's the metabolic region because this is where all the mitochondria are. So they are just making ATP or releasing ATP so that the sperm has the energy it needs to move. And then the tail structure is functionally known as the locomotor region because it has this flagellum that whips and propels the sperm and allows it to move. All right, let's talk about oogenesis. This is the process of forming female gametes. So it's going down in the ovaries. Females are born with all of the primary oocytes, so the early eggs that they're ever going to have, because this whole process starts in the womb in females. So meiosis in these cells begins in the womb, but then it pauses in late prophase 1, and it doesn't resume until after puberty begins, and that's when it'll make secondary oocytes. Secondary oocytes don't finish meiosis until fertilization occurs. So what's unique about this process is one oogonium cell is only going to make one functional gamete, one ovum or egg. The other 3 become these polar bodies. That's why you can see in this picture they're smaller, that basically just degenerate. So we said males can make 90 million sperm a day. Well, females are believed to only be born with around 7 million fetal primary oocytes. But by puberty, there's only about 400,000 of those actually left. And then again, you're only going to release 1 of those a month. So the math here, it just doesn't line up at all. There's way less eggs that are available than sperm. So those oogonia go through mitosis in the developing fetus. And then some of those will transform into primary oocytes in the fetus that are going to go through meiosis. But that meiosis pauses in late prophase 1. And then once you hit puberty, some of those primary oocytes each month will develop into secondary oocytes. But meiosis 2 stops in around metaphase 2 and is only completed in the cell that gets fertilized. So if a cell gets fertilized, then it completes meiosis. But if not, it never really even goes through this whole process. But again, we would have that S phase of interphase, meiosis 1, and then meiosis 2. All right, something we have to talk about in females are follicles. So each of those primary oocytes that females are born with are contained in an ovarian follicle, which is essentially just a cluster of cells that are all designed to nourish this one oocyte. So males have like one cystinocyte per sperm nourishing it. Females have a bunch of different cells nourishing them. So primordial follicles are the follicles in the fetus that are in the womb. That's shown like in number 1 of this picture. The majority of these remain dormant. They remain stuck in prophase 1. 99.9% of them are going to go through a process called atresia, which is just the apoptosis of the follicle and its surrounding cell. So again, I was telling y'all, you're born with about 7 million, primary oocytes in these primordial follicles. But by puberty, you only have about 400,000 left because so many of them have gone through atresia, and even more will. It's actually estimated, since you only release 1 egg a month, and that doesn't start till puberty, and then it ends, at menopause, that only 500 oocytes are actually ovulated in a woman's lifetime. 500. And males can make 90 million sperm a day. It's so crazy. So follicle development is happening along the same timeline and pace as the development of the oocyte that's inside of it. So after puberty begins, what's going down with these follicles? FSH, that follicle stimulating hormone, causes about 20 or less of these follicles to be recruited each month. So they're kind of saved from that atresia. And those develop into primary follicles, and then secondary follicles, and then vesicular follicles. So they're just developing. That's all you need to know. I don't care if you know each of these names. Okay, so you can see in picture 2, it's developing more. And then eventually, though, we're going to have one dominant follicle. Only one of maybe the 20 that get saved from atresia becomes the dominant follicle. Then the others are all going to go through atresia. This one dominant follicle will be ovulated. So see how it's going to be released into the fallopian tube. It's now a secondary oocyte in the dominant follicle, and it's the only one that's going to keep going through meiosis 1. And again, it's not going to go through all of meiosis 2 and finish it unless it gets fertilized. 6 through 8 show how this we form that corpus luteum, and then it gets degraded if pregnancy doesn't happen, which is something we learned about in the inquiry activity. All right, so let's compare these two processes of making sperm and making eggs. So total time to make one gamete, and for it to actually mature, is about 74 days in a male. So even though males can make 94 or about 90 million sperm a day, it's a continual process. But so each one's going to be taking about 74 days. Females, it's about 13 to 50 years, which is crazy. Okay, when is this going down? It's going down in males from puberty until old age. Females, it begins in fetal life, pauses until puberty, only continues until menopause, and then it's over. 4 sperm can be made from one germ cell through the meiosis process, whereas in females it's just 1 to 1. We're getting one large ovum, and then the other 3 are these polar bodies that degenerate. It's estimated that males make about 1 trillion sperm in their lifetime, whereas females are ovulating less than 500 that could actually become offspring. All 1 trillion of these could technically become offspring. This is also fascinating too. Error rate, so resulting in the wrong number of chromosomes being made, is about 5% in male sperm. It's about 20% in females. So even though they're technically probably ovulating about 500 ovum in their lifetime, a lot of those, a fifth of those, 100 of those are going to have errors in them, which would not even result in a viable potential offspring, which is kind of fascinating. Okay, so we've made egg and sperm in these semi-complicated processes. How do they get together? Well, that is fertilization. And that's when a sperm's chromosomes combine with those of an oocyte, and we get a fertilized egg, which is what we call a zygote. How did those get together? Well, those got together during copulation or sexual intercourse, typically. Obviously, this can happen artificially and in vitro fertilization. So, essentially, when an oocyte gets ovulated, it gets released. It's viable for about 12 to 24 hours in the female reproductive system. A sperm that gets to the female reproductive system through ejaculation, it can live about 5 days in the female reproductive tract. So even though a female is only technically ovulating once a month and that oocyte's only able to live there for about 12 to 24-hour window, there's technically a 5-ish day window that a couple could get pregnant if they're having sex in that window because the sperm can live in the reproductive tract for that time. Fertilization is, of course, most successful if it's happening, 24 to 48 hours after ejaculation when we've got fresh sperm in the fresh oocyte, but just know that that is possible. So after ejaculation, there's millions of sperm in the semen that's in the female vagina. Millions of those get killed just in the acidity of the female's vagina environment. Only a fraction are actually going to get through the cervix and into the uterus, and then even less than that are going to make it to the fallopian tube. So sperm has a journey. It has to reach the ovulated secondary oocyte in the fallopian tube before it can fertilize that egg. So it's got to get all the way through the vagina, through the cervix, through the uterus, and through the fallopian tube. And it's competing with all these other sperm in the semen. Then once it gets there, notice we've got like 4 sperm here that are around this egg. Only one's going to get in. A process called capacitation has to happen. It's a biochemical process that basically makes changes in the sperm so that it can actually get through that oocyte and actually fertilize the egg and hopefully make a zygote. So it's a long journey for the sperm and it's a complicated one. But it only takes one to fertilize that egg and get the zygote and potentially make a baby, which we will talk about in concept 3.